Hello, in this session we'll discuss more about the nature of drugs as chemicals. In particular, we'll talk about their permeation, their entry into the nervous system and into the brain. And then we'll spend a few minutes talking about a particular protein drug, botulinum toxin. First, routes into the nervous system. Nicotine enters the nervous system and then the brain via one of several routes. It can be smoked, which is the way about 1 billion people around the world get nicotine. It can be chewed, either as tobacco leaves or as nicotine gum. And it can enter through skin patches as well. Many people use nicotine patches as a way to stop smoking, which is an important goal. Procaine and other local anesthetics are generally injected into the sites where one wants to dull pain. Dentists use procaine a great deal, so do other physicians. Some local anesthetics related to procaine can also be put into creams, for instance for sunburn relief, and in the creams they can diffuse into the skin and cause local pain relief. Morphine can be smoked from the opium poppy. It can be injected by the physician or by the drug abuser, and it is also fairly common to bring morphine into the body via a suppository. So each of these ways is appropriate for morphine. And finally, botulinum toxin, when used therapeutically, is usually injected. When one gets botulinum poisoning, that's because one typically eats a food that's contaminated with botulinum toxin. So there are various routes into the nervous system. Let's discuss permeation through membranes and through lipid barriers. This is a key concept in drugs in the brain. When we're going to treat a general drug, an alkaloid, as having an R group, and these R's can be many groups as we saw in previously. So R is a general group, and we'll concentrate on the amine group. This is a primary amine, NH2 group, contained within the drug. This is a neutral form of the drug, and we get the neutral form of the drug by taking it in through the mouth or the stomach or the lungs, as in this picture here. Neutral forms of drugs are quite permeable through lipids, through fats, especially through the membranes. And so typically neutral forms of drugs go through membranes quite easily. However, it is the active charged form, which is usually the form that interacts with the drug receptor. So the neutral form takes up a proton, becomes protonated, charged, and this is the form that interacts with the receptor. This protonation, deprotonation takes only a millisecond or so, and it can occur either in the blood or the brain, or it can occur outside the blood and the brain, in the lungs or the stomach or the mouth. So that's a general statement about alkaloid drugs and about synthetic drugs as well that contain amines. And in general, when we have higher pH, this tends to take the proton off the charged form of the drug. So we have the neutral form of the drug. And so in general, higher pH at a site in the mouth or the stomachs or the lungs, a more basic solution, gives us more of the drug in the neutral form, which is able to permeate through membranes. Let's take as an example nicotine's path from the lungs to the blood and the brain. Tobacco leaves are roughly 5% nicotine by weight. So the vaporized nicotine, for instance, from smoking, is a neutral form. It has the N, now it has the CH3, so it's a tertiary amine. It gets through a total of 3 cells and 6 membranes in going from the lungs to the blood and then the brain. 2 membranes in the alveolar epithelium and 4 membranes in the capillary from the blood to the brain. And again, we have the neutral form of nicotine permeating, as smokers know, within just a few seconds from the lungs to the brain. A very important process that takes place roughly 150 billion times around the world every day when a smoker takes a puff. In the brain, the nicotine molecule is a weak base, and so it can easily gain or lose a proton, again in milliseconds. Its pKa is 8. That is, at pK8, it is roughly half protonated and half deprotonated. And it is, again, the protonated, charged form of nicotine that interacts with nicotine receptors. The same kind of equilibrium occurs in the lungs as well, and the cigarette manufacturers know this, and so they put ammonium hydroxide in their cigarettes to maintain neutral pH or to make it basic. The result is that the proton leaves the nicotine, and the nicotine is able to be more in its permeant form and permeates through the cell membranes. So that is the path from the lungs to the blood and the brain, taken by nicotine in just a few seconds, and it obviously depends very importantly on having a neutral permeant form of nicotine, even though the charged form is what interacts later on with the receptors. So if we look at the concentrations of nicotine in the blood during and after a cigarette, nicotine enters the blood, and from the end, it's probably even faster than shown on this slide, although it's difficult to measure it terribly fast. But within a minute or so, nicotine appears in the blood and in the brain, and then it rapidly gets metabolized, gets broken down. We'll talk about enzymes and other processes that break down drugs in the body in a later session. Here's another example of neutral drug permeation. It has to do with Parkinson's disease. In Parkinson's disease, most of the neurons that make dopamine degenerate, and so the challenge is to replace the dopamine in the brain. Dopamine, however, does not enter the brain. It's charged. As a result, physicians use levodopa or L-dopa, which is zwitterionic. It has no net charge and easily permeates through membranes, gets into the brain, where it is decarboxylated by an enzyme. Remember that an enzyme is a catalytic protein. So this deep carboxylase gives us dopamine, which can then, in the brain, which can then be taken up by neurons. So this is a prodrug, if you like, a molecule that is not itself a drug, but is a precursor to a drug and is made into a drug by the body's own enzymes. Now, the blood-brain barrier is a special structure made by cells lining the capillary wall called endothelial cells. The capillaries, the smallest blood vessels, come in 2 forms. In the periphery, outside the brain, the capillaries have spaces between the endothelial cells, see this space here, through which a large number of molecules can diffuse. Proteins, nonpolar molecules, polar molecules such as glucose. But in the brain, as we'll see, there are tight junctions, seals between the endothelial cells. And as a result, molecules cannot leave the capillary and go into the brain unless they're small nonpolar molecules. So only small nonpolar molecules can diffuse out of the capillary. Here's another view of the endothelial cells forming the blood-brain barrier. We have a capillary surrounded by endothelial cells with the tight junctions. And here are the red blood cells in the capillary. It used to be thought that the glial cells form the blood-brain barrier. They have feet. These are glial cells, astrocytes, which extend end feet around the capillary. It used to be thought that glial cells are the basis of the blood-brain barrier, but now we know it's endothelial cells. The structural basis of tight junctions is a structure called, the structural basis of the blood-brain barrier is a set of proteins called tight junctions. So here are two epithelial cells. One is here, one is here. They each have double layer membranes. In these double layer membranes are proteins that make up these so-called tight junctions, and you could think of them as zippers that zip together the two cells that make up an epithelium or an endothelium. So we have one extracellular solution up here, perhaps the blood, another extracellular solution down here, perhaps the brain, and the tight junctions occur only in the brain capillaries and prevent molecules from diffusing through. Drugs in the body and in cells are an important topic in drugs and the brain and in new development for medications. There is acid-base equilibrium and permeability, which we discussed, protonation and deprotonation. We've discussed uptake from the stomach, uptake from smoke, crossing the cell membrane. There's another topic, short-circuiting of synaptic vesicles. We'll discuss that topic later in later sessions, and likewise we'll discuss neurotransmitter transport inhibitors in later sessions. We've discussed the blood-brain barrier and a bit about its molecular basis. Clearly, the fact that the blood-brain barrier allows molecules to work in the periphery but not in the brain is an opportunity for drug specificity, and many drugs utilize that opportunity, but it's also a problem for drug delivery. And how to get around the blood-brain barrier is going to be a very interesting problem over the next few years as we try to develop better drugs for the brain. We'll stop here and go on to botulinum toxin in another session.